All right, so yeah, we're really excited to be here and be invited to share about the assessment that has been designed to measure chemistry knowledge and scientific practice proficiency using argument-driven inquiry as a pedagogy. So we've seen a lot of conversations, and there's been a lot of conversations over time about what we should be utilizing the chemistry lab for. We have this really unique space for students to practice whatever we want to have them practice in terms of chemistry. And so at East Carolina University, we would say that laboratories should be utilized for students to develop proficiency in practicing what scientists do. So we want them to develop proficiency with scientific practices. So what we mean by scientific practices, we're using Ford's description where he describes this dual nature of the practices of science. So there are two distinct but complementary components where the first types of practices are where you're physically manipulating nature to study the aspects of it. And the second is when you're using practices to actually communicate science to peers. So you're making nature's behavior apparent to peers. And more specifically, Ford described two types of practices. So here in purple, the empirical and representational practices. So these empirical practices are when you're physically manipulating matter, you are planning investigations, you're collecting data, you're analyzing that data. And the representational practices are when you're taking what you've collected and developing models, visualizations, crafting arguments to communicate what you found. There's also interactive practices where you're proposing and supporting ideas, you're critiquing and challenging ideas and revising them based off of your data that you've communicated. And these are the types of things that we want students to be practicing in the laboratory. And the way that we have them practice scientific practices at East Carolina University is through a pedagogy called argument-driven inquiry. And so argument-driven inquiry, instead of having students perform unique experiments every week, students perform investigations that last over 2.5 class periods, excuse me. And so we have students in collaborative teams of 3 to 4. During the first class period, they'll participate in what we call a skill builder activity. So they're developing skills that they'll need to have to continue on in the investigation. So things like titration, how to read volumetric glassware, those psychomotor skills. In the first week, they're introduced to a guiding question, and then they have to create design an investigation that they will perform to specifically address that guiding question. And this is all done in that first week, in that first class period of an ADI investigation. In the next course period, they will collect data and analyze it according to the design according to their investigation design. And they'll participate in an argumentation session. So using the data that they've collected and analyzed, they will create an argument that they will then share with their classmates and edit their argument as they see fit based on what their classmates have told them. And this happens in the second week of an investigation. Between the second week and third week, students write a lab report, which I'll show you more details in a second, but they bring that lab report to class in week 3 and they participate in peer review with their classmates. And then this peer review doesn't take the entire class period. So they'll start week 1 in the second half of that third class period. So like I said, an ADI investigation takes 2.5 class periods. After week 3, they will take their peer-reviewed lab report and revise it and turn it in digitally in that 4th week. So this is a complete ADI investigation schedule. And I described Ford's description of scientific practices, and I want to note that ADI was being developed at the same time as the science and engineering practices were being developed by the NGSS in America. So the science and engineering practices are used nowadays heavily in K through 12 schooling as a way to assess, what students are doing in science. And we use it oftentimes in higher ed as well. And so these are very similar to what Ford described, such as, asking questions, constructing explanations. engaging in argument. And I just want to point out that the way that ADI is structured still aligns with the NGSS science and engineering practices. And so this is an important thing to note. So what does this look like in class? So first, students are given a guiding question that they have to then design an investigation around. And so the worksheets look something like this, where this is the second ADI investigation that students do in Gen Chem. So the guiding question is, whoops, excuse me, what are the products of the chemical reaction? They're given two different hypotheses, so two different products that the chemical reaction could produce. They then have to come up with a procedure. So what type of data will you collect to address the guiding question? How will you analyze the data, and what safety precautions will you follow? And then their predicted results for hypothesis 1 or 2. And I just want to point out that not every ADI investigation has a hypothesis associated with it, but this one specifically does. So this is given to students in the first week, and they complete this in the first week of their investigation. During the second week, they perform their investigation, they collect their data, they analyze it, and they come up with this argument that they write on a whiteboard. So at this point, they have analyzed their data, they write the guiding question on the whiteboard, they then write their claim. So in this case, what were the products of the chemical reaction? They write the evidence that they've collected that day from their designed investigation. And then they write their justification, which connects their evidence to the claim. So why was your evidence appropriate for this claim? And they write this on a large whiteboard and they put it up at their lab station. And so, like we said, there's 3 or 4 students who have worked collaboratively to create this argument. And then they participate in an argumentation session. So one of the students will stay with their whiteboard that they created. So in this case, it's the gentleman on the right. He's the presenter then, who's presenting the argument that his group came up with to students who are traveling around the classroom. And so this rotation happens about 2 to 3 times. So students are seeing their classmates' argumentation, sharing with each other what they found. And at the end of the argumentation session, they reconvene and share with each other what they learned from their classmates. And at this point, students can revise their argument as they see fit, according to what they learned from their classmates. So after the class period where they participate in an argumentation session, students write an investigation lab report. So I mentioned that this isn't a formal lab report. We have them write 3 separate sections. So the first one, we ask them, what concepts were you investigating? So we have them relate the concepts to their guiding question. The second section is, how did you go about your work and why? So discussing their design process and justifying why they collected data the way they did. And then section 3 is, what is your argument? So they have to justify the data used as evidence for their claims. So they have to provide their claim evidence and justification. And so throughout this entire process, we have students participating in scientific practices. And so how do we assess that? Well, at the end of the term, we have students participate in the investigation, design, explanation, and argument assessment, or the IDEAA assessment, because that is a mouthful. And this is an end of term practical. So students are physically coming into the lab at the end of the term and completing this practical. And the practical is designed after an ADI investigation, and it's designed to assess scientific practices. And so in this case, the IDEAA for General Chemistry 1 has 2 parts. And the first part focuses on arguing from evidence, and the second part focuses on planning and carrying out an investigation. And so this leads us to our research question, which is, how do students perform on a practical design to measure content knowledge and scientific practice proficiency? And so the way that we assess students, we gave this idea in General Chemistry 1, where there are 48 students per section. It was administered at the end of spring 2019. So this is the off-sequence General Chemistry 1 course. Students are given the full lab period, so they're given 3 hours to complete the practical. And students in the course are a plurality of sophomore level students, so second year students. And there were about 325 student practicals that we looked at to study student performance, which is about 87% of the total enrollment in 2019. And just taking note from Britland, I put where East Carolina University is in the United States. So they are on the East Coast of the United States. And with that, I'll turn it over to Joy to talk about the first part of the idea.
Okay, so this practical was developed as part of a larger project where we were implementing ADI into not just chemistry, but biology and physics. And that's going to be important towards the end. So as you can see, we were giving the students an experiment set up much like an ADI. They have a guiding question. They're given a problem, which is also part of the documentation given to students in ADI labs. Then here we gave them the procedure. They didn't provide the procedure. And I want to say this was not an exact experiment the students had done. They had done the chemical reaction one that Katie talked about in the intro, but they had not done a combustion. But this was still a reactants and products. And the questions that we asked are similar to what we had done in lab. So we gave them this, and then we gave them a student notebook page, which had very succinct data. I realized there should be replicates, and the paper we published over a year ago now, goes through the iterations we had. We tried having replicates, trying to implement this in 15 sections and ensuring gradability, reliable gradability with all the grad students. We needed to minimize distractions, let me say that way, and having multiple trials kind of made things a little messier than we wanted. So this was the data they were given, and then they are given a answer sheet to complete. Whoops, I jumped ahead. So all of our, you can go ahead, Katie. I was going to the answer sheet. So we use the anchoring concepts. This is out of the ACS. Katie's, we've cited Tom Holmes here. They have gone through the anchoring concepts and the ACS exams and identified a significant number of science practices that are represented in these. So the anchoring concept, I'm just going to, with these, talk about level one, this level number 5 anchoring concept is very much about chemical reactions, how chemical reactions occur, drilling all the way down to the content details of balancing equations, stoichiometry, calculations. So this is really encapsulates the first part in terms of the content knowledge that we're expecting students to apply in these science practices. Now we can go to the answering sheet. So here I've aligned the answers that we were asking the students to give with science practices. So the first 3 questions are analyzing and interpreting data, and this is where they were looking at the information we had given them. Question 4 is developing and using models. So, chemical equations are a model for what we say is happening in the flask. So that is the practice there. And then the stoichiometric calculations and the limiting and reagent calculation, theoretical yield calculation, those are mathematical and computational thinking. All of that was necessary. And I can tell you my original draft of this, because I was a researcher, did not have all of these steps and really just went, here's the data, write me an argument. And again, if you look at, if you're interested on the iterations that this went through, you can read the paper from a year ago. But ultimately, the practice we were looking at here is constructing an explanation and arguing from evidence. So now let's look at the results of this. So part 1, analyzing and interpreting data. They all managed to subtract the dish. And I'm going to real quickly, one of the things, this is not a multiple choice exam. So we had a rubric and, we had incorrect, correct, and partially correct. And I think, again, this is very important in terms of student understanding what students do. We have them, write out answers. And so, it's not clear, you don't want to just say right and wrong. There's stuff that's kind of in between there. So we had a rubric for that included a partially correct. So The product color, they should have noted in the results in the student notebook page that upon heating, the copper compound turned black. We had, some mixed values with that, but again, 88% got some part of that correct. And then the other element present, this really was a two-part question. They had to first identify oxygen and then also say the oxygen was from the air. Sometimes they identified monatomic oxygen as the elemental symbol. We accepted that. Understanding that the oxygen was coming from the air was probably our biggest issue in terms of partial credit here. Okay. The balancing, again, we tried not having, we kind of prompted the balancing when we just left it blank. And said, write a balanced equation. That led to just too much variety of results. As a colleague of mine said, it was very interesting to me as a researcher, but in terms of an educational process, it was too complicated for grading and scoring effectively. So balancing equations, they pretty much put O2. If they got partial credit, it was monatomic. They put the copper 2 oxide as the product. The second hypothesis is actually harder to balance, and so that explains the difference there. And then finally, the theoretical yield. And of course, what we're going to see here is what is being published, in fact, was just published last week in SERP by people looking at issues with stoichiometry and Gen Chem. Mole ratios, they can do molar mass. The mole ratios were kind of all over the place. They could set up the dimensional analysis, but when it came to actually doing the calculation, dimensional analysis just doesn't seem to work, even though we keep beating that drum. So this again, I think, just establishes something that's been well established, but that is still used, pervasively in Gen Chem. So here's our argument, which is the goal of all of this. So they had to make a claim, and then they had to use their evidence, and then they had to justify their evidence. So again, this is exactly as they've been doing in lab all semester. And we see fairly good claims. The use of evidence is a problem, and I have some examples that I'll kind of show what was going on with that. It's really that they don't ever use all their evidence. So, and then the justification, and I've been doing work on argumentation for 15 years now. Justification is hard. Getting the whole justification in there. It's not even a skill that's developed until late high school. So freshmen in college are still getting the hang of justifying their argument. But these were pretty good. We were pleased with these results. So next.
I also just wanted to add that we may not need to have students be able to completely do justification right, because it's a practice. So so this is good, right?
Yeah, we're learning. It's Gen Chem 1. These are novice learners, despite the fact that the law says they're adults. So here are two examples. The first one, right away, you can see there's 3 numbers in there. So they used all their evidence. So they got a 2 for evidence, but they never explain, right? So they say the theoretical yield is much closer to the final amount of product, meaning the powder is in the unmarked bottle is most likely copper 1 oxide. So that, that's a 1 justification. They don't tell me why closer counts. Why does the theoretical yield and the actual yield matter? Why do those count? So that was an issue with that one. The second one is sort of the reverse. And again, you can look at this, see they used 1 number. And this is something that is very important in terms of argumentation, is making sure students are explicit with their evidence, because we don't know what theoretical yield, right? They say the physical yield of the experiment was 1.85 in comparison to the theoretical yield. Well, I don't know what number they were using, right? So I have no way of knowing what yield they were using. So that was a 1 for evidence, but then their justification is actually, spot on. Actual yield cannot be more than theoretical yield. So those are the two examples of sort of the two cases. So for GC1, part one, analyzing and interpreting data, the issue, and this, we're kind of evaluating our assessment here. They were 75% proficient at interpreting data. The source of oxygen and air was problematic, which I think that's problematic like on a much bigger level than general chemistry. We really need to drive that home. Developing and using models. Again, 75% proficiency, but trouble writing chemical formulas and balancing. I've already talked about the issues with the mathematical and computational thinking, and then with the construction of argument from evidence. This is just incomplete. Justification is hard, and realizing that they need to use, fully justify, explain to me why actual and theoretical yield count. So those were the issues with part one. And Katie's going to talk about part two.
Yeah, so in part one, we give students that example of an experiment or investigation, and we write out the procedure for them. In part two, we have them come in when we give them this prompt, which talks about how KHP can be used as a standard to standardize sodium hydroxide. And we ask them to do a titration by themselves, design the titration and carry it out by themselves. We tell them that the flasks of sodium hydroxide are approximately 0.1 molar, but that they need to determine the exact molarity of their unknown. So for part two, the content knowledge that is being focused on are, experiments, measurement, and data. And so just pointing out these content level details. So understanding the techniques or being able to perform techniques to set up and carry out a laboratory experiment, to be able to determine solution concentrations, usually with titration, and then understand the errors in measurement throughout that process. The scientific practices that are focused on, so like we said, planning investigations is the main thing that we're focused on, but we also are measuring using mathematics and computational thinking. So we ask them to first describe in detail the procedure that they'll use and create a data table to record their data. And then we ask them to actually calculate the molarity that they found using their titration and record their molarity. And so how did they perform on this? Well, on the first two questions where we have them plan an investigation, there were several things that were included in the rubric for these two questions. So first of all, with designing their procedure, 34% of students actually wrote a complete procedure and how to perform a titration, just walking into the room, reading that prompt and writing a complete procedure on how to carry out a standardization of sodium hydroxide. This 27% of students that were a little bit of points were taken away because they didn't include certain details like properly preparing the burette or removing the funnel after the solution is added. So over 50% of students were able to come in and create a procedure to carry out a titration by themselves. The next three items are just them recording the appropriate data. So you can see a high percentage of students recorded the appropriate data that they needed when they were carrying out this titration. The volume recorded to 0.01 milliliters. This was an issue for, 50% of students did record to the correct milliliter digit, but 50% didn't. And this was a focus of the skill builder lab where they were taught how to read a burette. And so this points out to a larger issue of students understanding the difference between a value and a measured, a number and a measured value. And so on making sure they understand why they need to read to a certain milliliter is something that we can focus on. And then the accuracy of the molarity was calculated using, so the TAs would calculate what students should have gotten according to the data they collected, and then judging their value based on that answer to the thousandth digit. And so, over 50% of students, 70% of students were able to come in, perform a titration, and get a fairly accurate molarity of their standardization of sodium hydroxide. Next is just an example of a data table that a student created while performing this investigation. So, this isn't a data table that maybe we would write, but Ford emphasizes that students need to be able to, organize their own data in their own way when they're learning. And so you can see that they have all of the necessary data organized in a certain way. So they've recorded the grams of KHP for each iteration, and then the initial and final sodium hydroxide measurements, as well as the observations of the phenolphthalein at the endpoint, which is an additional observation that they added. As far as using mathematics and computational thinking, we saw really similar things from what we saw from when this was measured in part 1, where students basically have their dimensional setup. Their dimensional setup is okay, but it's the algebra that's holding them back. In this case, the mole ratio of KHP to sodium hydroxide was 1:1. So students weren't necessarily showing that they understood that. So a lot of the students did get this incorrect because they weren't showing that it was a 1:1 in the way that we were assessing them. They were forgetting to convert milliliters to liters, and the significant figures, the appropriate amount was 3, and many of them weren't getting that correct because of the way that they recorded their volume when reading the burette. Like I said, they may not have recorded to the appropriate amount of significant digits. And so again, this just points to the fact that in this way, we need to kind of reconsider either how we're assessing them or how to focus on this type of analysis, because in this way, we're focusing on algebra, and we know that that's an issue for students. So to conclude part 2, when asked to plan and carry out investigations, 60% of students were at least partially proficient in coming into class, performing a titration from memory, writing out that procedure. And over 70% of students were able to complete that titration and receive a somewhat accurate final molarity of that solution. And then again, just stating this again, that students have trouble with algebra in this mathematics and computational thinking. So we need to kind of rethink how we're asking them to do this science practice. As far as limitations of the overall IDEAA, the IDEAA is aligned to ADI specifically. So this is specifically aligned to a pedagogy that implements science practices. So the assessment is mirrored after it. And for researchers, these are results from one institution. We're still revising the IDEAA. And so we would need to investigate validity and reliability in additional settings if we wanted to use this assessment for further research. And now I'll kick it off to Joy to end things. Oh, you're muted.
Thank you. So as I mentioned, this was part of a broader project. And one of the points I want to wrap up here with, which has been brought up, Britlin said it, is that assessment drives what we do and what students consider valuable. And the fact that we were able to show in the ADI labs and with the assessment we developed for chemistry that they could do it. Because believe me, I was told the students couldn't possibly, there was no way they could come in there and do a titration. Couldn't happen, right? So 72% of them came in and did a great titration. So biology and physics then took this same idea, because they had to develop a similar investigation. And whereas we had 2 parts, they just had them, come in and do an experiment. So the biology experiment, they had the choice of looking at the best methods to store lactose using heat, pH, or an enzyme. Physics, they were studying rotational motion, don't ask me any physics, which hadn't been taught that semester. So their investigations or their practicals were a full investigation. So I want to show the results from those. So this is just the argument question from the first semester labs. And I just can't tell you how much the pandemic destroyed this study. I can't even, because I'll just have to have therapy. So if you look at, and what I want to particularly focus on is, you remember in chemistry, we saw them not really using their evidence, but look at how much more they're using evidence in physics and biology. And I really think that has to do with that they found the evidence themselves. They weren't trying to interpret someone else's evidence. The justification is still problematic, but not quite, about the same in biology and chemistry, a little worse in physics. But their claims were all spot on. And I just always, if you look at the claims that they made in physics, biology, those were even stronger claims than we saw, more students successful in their claims. So I'm just always impressed with this, because this was an investigation students walked in and just did. So, so my broader impact point here on this method, because I want to say, this was developed in 2015. You can go ahead and tab down. I had to use it for 3 years because I had a grant. We were trying to collect data. So the first thing I want to say is that measuring science practices is always going to be confounded by content knowledge, by what they know, right? Or not always. I mean, you could have them just do a practice, but if we're going to try and do this assessment, the two are conflated. I also want to point out practicals are practical. I mean, people, even now I get people say, oh, we couldn't do that with, our sections are too big. I mean, we have 15 sections. It can be done. Chemistry provided evidence that that showed what students could do, that showed they could do this. And so it has broadened the assessment potential, not just in physics and biology, but up the chemistry curriculum as well. So even though, like I said, this was developed in 2015, the grant's over, so we're making changes to it. It's far from a perfect assessment. But what we've tried to do with this is kind of show you the limitations, but also show you that we could measure science practices within content knowledge. So here's where we are now. We now have ADI. I can say that at ECU, there are no cookbook labs at the intro level, none, 0. They are all ADI inquiry-based labs. We do a cross-disciplinary GTA training, which is my favorite day of the year. We have grad students from 5 disciplines that we work with all day. We get to indoctrinate them into active learning and such things. We have expanded in biology. They're now in chemistry. We're now doing expanded in second semester, more project-based, because they're ready for it. We're doing an amino acid titration. But I think our biggest impact, and thanks to Gwen for giving a shout out to me and Katie this at 5 a.m. We are running cures. I can't keep up with the cures, right? And some of it is we did similar assessment in the chemistry cures, and we're able to show student learning in chemistry cures, and those are published. Next fall alone, I have 14 cures running in 4 disciplines. So I think the broader impact of having an assessment that really measures what students can do and know, I can just say for our context, it had a profound impact. Here's my group on the right. They're all gone, sadly. On the left are my 2 new friends working with me, I'm super excited about. Thank you to the NSF and to ECU, and to Katie Hosbein, who has gotten a assistant professor job. Anyway, here's our emails, and that's it.